And so in graduate school, it seemed like many of my questions were answered. I came across this biological sensitivity to context model. It was proposed by Tom Boyce and Tim Ellis. And what it basically argues is, that people possess these individual characteristics that influence how they respond and their sensitivity to different environments. There's sort of this, I'm not sure how familiar everyone is, there's something called the Orchid and Dandelion hypothesis. If anyone has kids, you might relate to this. There's sort of the Orchid kid is a kid that might be equated to a kid with a difficult temperament. So there's someone that's very, very sensitive to environmental variation. And then there's the dandelion kid that's sort of fine regardless, similar to a dandelion plant, sort of fine regardless of the environments that they're in. And there's two competing models sort of within biological sensitivity to context. So the first is the Diathesis-Stress Model. And the Diathesis-Stress Model essentially suggests that people have these individual vulnerabilities, and that when these individuals are exposed to contextual hardships or challenges, we see these sort of significant dips in functioning. So for example, here we have individual A and individual B. Individual A does not have the attribute that confers risk for impairments, right? So we see that individual A, even though they're exposed to this negative environment, their functioning remains relatively constant. But then we have individual B that has this characteristic that makes them more likely to experience challenges when exposed to contextual adversity. So then I came across this differential susceptibility hypothesis. It was proposed by Jay Belsky and Michael Cluse in 2009. And this theory essentially builds on the Diathesis-Stress Model. And the idea is that what if people don't possess vulnerabilities per se? What if they're actually susceptibilities, right? So what if people aren't just sensitive to negative environments, but to context more generally, okay? So you have individual A and individual B. Individual A basically possesses characteristics that do not make them more sensitive or vulnerable to environments. And you can see that when they have this negative exposure, they're not affected. When they have this positive exposure, they're not affected. Their functioning remains relatively constant. And you have individual B. They have this vulnerability or susceptibility feature of some sort. When they are exposed to a negative environmental exposure, you have this significant dip in functioning, right? And you see this, that it goes below that of individual A. But when they're exposed to these positive environments, you see this significant elevation in functioning above and beyond that of individuals that are less susceptible, right? And so this was very interesting to me. I was very excited about this, and for years I focused on testing these two different models, right? And so I was specifically interested in temperament. Many people so temperament, we're talking about individual differences in reactivity and self-regulation that influence things like cognition, affect, and behavior. And it's often considered a proxy for sensory processing sensitivity. And the idea is that, kids with difficult temperaments, for example, we know that when they're exposed to poor parenting or sort of poor quality neighborhoods, they generally do worse. And we're talking about worse, they evidence higher levels of psychopathology. But the question is, are they going to do better? Would they do better than their peers that have easier temperaments, right? And that's what I was really dedicated on studying for quite a long time. And so this is just an example of some of my findings. So in this study, I looked at the intersection between temperamental flexibility, so one's ability to adapt to changing environments, and parenting in relation to externalizing symptoms, essentially whether or not one acts out, is sort of delinquent or aggressive. And so what I found, so this is kids low in flexibility and high flexibility. And so the low flexibility kids, when they were exposed to low positive parenting, they evidence higher levels of externalizing symptoms, right? So they're more likely to act out in these poor and negative environments. The shaded gray area reflects the statistical significance between the two lines. So what you can see is that among these low flexible kids, you do see this attenuation in symptoms as the environment improves, right? But they're never doing better than the kids that are high in flexibility, right? So this is evidence and support for the Diathesis-Stress Model. So again, while I was very excited about the prospect that maybe kids with difficult temperaments might do actually better, I did not find that in the work that I've done over the years. But this work did inspire me to the work that I'm currently doing, which is focusing on genetics. So temperament, its behavioral manifestation, it's largely influenced, it's seemingly a byproduct of endophenotypes or biomarkers, but again, it's also heavily genetically influenced. So there's a number of ways that one can study genetics. This includes twin and adoption studies, molecular genetics work, as well as a candidate gene approach. The method that has been very sort of commonly used and has been supported substantially by empirical and theoretical work is that of a polygenic risk score approach. So polygenic risk scores, they are created from genome-wide association studies. And genome-wide association studies attempt to identify genetic variants associated with a given phenotype or outcome. And typically you'll have something which is called a Manhattan plot produced from a genome-wide association study. So just to orient folks to this, so this is the x-axis reflects chromosomes. You have 1 to 22 as well as the sex chromosome. And this essentially, the y-axis reflects the threshold for significance. And so you can see that there are a number of genetic variants that have reached this threshold for significance and that are implicated in being a self-reported morning person. So you can think about your own statuses and whether or not you're a morning or a night person, and maybe you have some of these genetic variants. Who knows? So as I said, these scores are created from genome-wide association studies, which attempt to identify single nucleotide polymorphisms associated with disease. So what is a single nucleotide polymorphism? So a single nucleotide polymorphism, or a SNP, refers to variation in a nucleotide. There are 4 nucleotides, A, T, C, and G. So in this example here, excuse me, we have...
It doesn't work on that.
Oh, it doesn't work? Okay, that's fine. That's okay, that's okay. So we have 2 strands of DNA. At this specific site, this person has an AT, and for this other person, this person is a GC. So this is what we commonly see. This is just simple genetic variation, okay? Okay, so when we are attempting to evaluate risk for disorder, as I said, we do this GWAS, and results from a genome-wide association study will typically give us an allele or genetic variant that's associated with a given disorder. So in this case, say we're trying to evaluate risk for autism spectrum disorder, and the GWAS indicated that the T allele was the risk allele. So individuals with the genotype AA are at 0 risk. They don't have the alleles. Individuals heterozygous for the T allele, they have 1 of the alleles, and individuals homozygous for the T allele, they have 2, right? And so what you do is for each individual, you identify at each specific SNP site whether or not they have the risk allele. And then you sum up at each SNP site whether they have the risk allele, and you get a summed risk score. Now, results from a genome-wide association study, you'll not only get the risk allele, you'll also get a measure of the effect size. To what extent is this genetic variant associated with risk or resilience for a given disease or outcome? So at a given site, the T allele is associated with an increased risk for a disease of 1.2 log-ons. Individuals with the homozygous for the A allele, they have no risk, they're completely fine. Individuals that are heterozygous for the T allele, at this specific site, they have an increased risk of 1.2 log odds, and individuals that are heterozygous for the T allele, they have an increased risk of 2.4 log odds of ASD in this case. So you have, again, the GWAS produces effect size for each specific site and each specific allele. So these are your effect sizes. You identify at each specific site whether or not the person has the risk allele. You multiply that by the odds ratio. You sum up all those values, and you get what's called a weighted polygenic risk score, which essentially reflects one's genetic burden or genetic resistance for a given disease or outcome. Does anyone have any questions? So just to go through the steps, you have a genome-wide association study, and this is typically called your discovery sample. And in this sample, you identify the top SNPs and risk alleles associated with given disease. Then we take results from the genome-wide association study, or you take the genetic variants, and you apply them in your target data, right? So you select those SNPs in your target sample, and you evaluate whether and to what extent the SNPs identified in the GWAS are associated with an outcome in your sample, or what's typically called the target data. So here's just an illustration of polygenic risk scores in action. So this is looking at the likelihood of developing schizophrenia. You can see here there's a really beautiful linear relationship between number of risk alleles for schizophrenia, as well as one's odds of developing schizophrenia. So the more genetic variants you have that are associated with schizophrenia, the greater your likelihood is of developing schizophrenia, right? And in a lot of cases, the amount of variance accounted for by these scores, it really, really varies. Generally, the larger the sample size, and if a disease isn't, well, I guess it depends. But the larger the sample size, you will see a greater amount of variance accounted for in a given outcome, which I'm going to talk about in a second. So what most determines the extent of PRS prediction? Sample size, as I just discussed. Another important factor to consider is whether the phenotype from the genome-wide association study matches the phenotype under consideration in your target data. So I do a lot of work looking at, for example, internalizing and externalizing polygenic risk in relation to substance use disorder. Naturally, you would expect to see some level of attenuation in the variance accounted for if the original GWAS of internalizing, externalizing symptoms, we're focusing again on a slightly different phenotype. One thing that really, really matters is ancestry. Ancestry is incredibly important, and when we're talking about ancestry, we're talking about differences in family lineage. So here's just an example of how ancestry can matter and shape genetic differences that we might see today. So here is a map, and it focuses on malaria transmission areas. So in the dark purple, here's an area where malaria transmission is very, very high, and in the lighter purple, it's a bit lower, and in the green areas, essentially, there's virtually no malaria. There's a genetic variant called the HBS allele, excuse me, and it's been shown to be protective against malaria transmission. And what you can see here in a very sort of clear way is that in regions where malaria transmission is high, individuals are more likely to have this HBS allele, right? So, and it makes sense conceptually, because if malaria is something that kills you, right, the individuals that had sort of some genetic protection, they were more likely to survive. And so those genetic variants were passed on. So you can see here again how the environment and sort of geographical region can really shape genetic differences.
That's the sickle.
Sorry?
Hemoglobin S.
Oh, is it?
Yeah.
Oh, interesting. Okay, I didn't realize. Okay. state of genome-wide association studies in ancestry. So you can see here, so this represents the number of genome-wide association studies that have been conducted from 2006 to 2018. So you can see that across the board, there's been this increase in genome-wide association studies for a number of reasons. I mean, the cost of genotyping has gone down dramatically over the years. But what you can see is that most of the genome-wide association studies conducted is predominantly on individuals of European ancestry, right? Despite the fact that they represent a fraction of the global population. And you might be wondering, well, does this really matter? It turns out it really does. So here is a violin plot showing the level of prediction accuracy when you take genome-wide association study results from a predominantly European ancestry and you apply them to different populations. So you can see here when you discover results from a European ancestry, apply them to another European ancestry population, prediction is perfect, which is fantastic. That's really, really wonderful. But you can see that the prediction accuracy really declines, right? So when we're talking about individuals of African ancestry, for example, we see this decline in predictive accuracy by about 75%.
Ooh.
Yeah, and so in a lot of my own work, what I found is that we might see something that's predictive of, I don't know, in the original GWAS by about 10%. And then when I've been, with the work that I'm doing, I oftentimes see it accounts for about 1% of the variation. And that could be due to a number of reasons, but the main reason, in my opinion, it's just ancestry. You're missing the alleles. There's a lot that goes into it, but essentially it's a really, really big problem. So now I'm going to talk a little bit about the work that I've done looking at genetic and environmental predictors of alcohol use disorders. Oh, before I do that, just want to say I'm really interested in genetic and environmental interplay associated with development of substance use disorders, particularly in admixed and diverse ancestral populations. And now I'm going to talk a little bit about the work that I'm doing on the work that I've done looking at gene and environment interplay as it relates to alcohol use disorders. So in this one study, we were particularly interested in internalizing the relationship between internalizing polygenic risk and alcohol use disorders. So we know at the behavioral level that higher levels of internalizing symptoms are positively associated with alcohol use disorders. It makes sense conceptually that individuals that are more anxious and have higher levels of negative affect and distress might be more likely to use alcohol to self-medicate these symptoms. And there is evidence at the genetic level that internalizing symptoms and alcohol use disorders are genetically linked. Much of this comes from studies that take a candidate gene approach, and most of these studies are also focused on individuals of European ancestry. One contextual factor that is also robustly associated with alcohol use is community disadvantage. In disadvantaged communities, youth are more likely to encounter an adult using alcohol. They're more likely to perceive drug use as normative, which might lead them to use alcohol in these settings. In addition, in disadvantaged neighborhoods characterized by residential segregation, decreased supervision of youth, as well as increased concentration of alcohol outlets, youth might have just greater access to alcohol, and that might be more likely to develop an alcohol use disorder. And there's some evidence that the combination of depression or phenotypic depression, self-reported depression, and community disadvantage increases risk for alcohol use. There's some work that's been conducted in this sample that has shown that. Excuse me, but it's unclear whether disadvantage might influence the relationship between internalizing polygenic load and alcohol use disorders. And so the first question we sought to address was, is there a relationship between polygenic load for internalizing symptoms and alcohol use disorders? We expected the relationship to be positive. And the second question was, does community disadvantage moderate the relationship between internalizing polygenic load and alcohol use disorders? And we expected that in more disadvantaged communities, individuals with a higher internalizing polygenic risk score would be more likely to have an alcohol use disorder. So data from the present study was drawn from 3 cohorts of participants from the Prevention Intervention Research Center. The participants were originally recruited for an elementary school-based intervention trial, which sought to reduce behavior problems and improve academic achievement. Participants have been followed from first grade to young adulthood. Data are currently being collected on the participants who are now about age 30. We have about 1,300 participants that have genetic data with substance use data, and in the current study, we restricted the sample to only Black participants to reduce the likelihood of population stratification. Past year, alcohol abuse and dependence at age 20, community disadvantage was assessed via geocoding in first grade, and we created a polygenic risk score for internalizing symptoms based on a GWA meta-analysis conducted by Kelly Benke, which included a discovery sample of about 5,000 people. So our first question, is there this relationship between internalizing polygenic load and alcohol use disorders? It turns out not really. There was a marginally significant negative association between internalizing polygenic load and alcohol use disorders. We thought, well, maybe this relationship, the relationship between internalizing polygenic load and alcohol use disorders becomes more robust when we consider sort of the environment, and that's essentially what we found. Excuse me. So we found a significant internalizing PRS and community disadvantage interaction. So in particular, and this was a bit counterintuitive and not what we expected, individuals with a lower internalizing polygenic risk score had a greater likelihood of developing an alcohol use disorder in high community disadvantaged contexts. So we sort of thought about, if you think about internalizing and externalizing psychopathology on a continuum, they're in some ways related. And so we expected that, well, maybe folks that are really, really low on this internalizing genetic load, maybe these are the folks that are higher in sensation seeking potentially, maybe they're a little bit more impulsive, which might confer risk for using alcohol in these contexts. But it was, a bit surprising, but that's kind of what we suspected might be going on. And so the second question we sought to address is, we saw this trend level negative association for this internalizing polygenic risk score and alcohol use disorder at age 20. Are we going to see those same results when we're looking, when we're considering alcohol use from adolescence to young adulthood? And what does the role of community disadvantage play in the relationships we might see? So we used a subset of the larger sample. It's kind of tricky because you have to kind of determine, which participants have which data at what times. So the sample included about 400 African Americans. Frequency of alcohol use was assessed between the ages of 14 and 26. We assessed community disadvantage in 6th grade, and we used the same discovery results for internalizing symptoms that we did in the previous study. And so we found 4 different classes of alcohol use. So the first is the low steady class. So this was characterized by a group of young people that didn't report using alcohol from ages 14 to 26. Then you had, then we found an early onset decrease in class. And this was a group of individuals that reported using alcohol at age 14, sort of some increases in alcohol use up to age 17, followed by a attenuation in use. We also see a late onset moderate use class that started drinking around age 21. And then we also have a early onset increasing class, which essentially started to drink around age 14, and they see this significant increase in use over time. So what we found, so again, we were interested in looking at the relationship between internalizing polygenic load and alcohol use over time. And we found that relative to the low steady class, the internalizing polygenic risk score was associated with membership in the early onset decreasing class. So in other words, individuals with a higher internalizing polygenic risk score, they're more likely to be in this class that uses alcohol up until age 17 and followed by a decline in use. So one of the things we thought about when attempting to think about why these results were the way they were, we had thought, well, We know that early and middle adolescence is a time of transition and change for many young people, a lot of contextual changes, social and biological changes as well. And so maybe individuals with a higher internalizing polygenic risk score, they might have more difficulty adapting to these changes, and maybe experience higher levels of negative affect, and they might use alcohol to kind of attenuate some of the negative emotional affective states they experience. It could be that age 17, sort of later adolescence, there's some negative repercussions of using alcohol that these folks might be more sensitive to, which might result in sort of that declining use that we see here.
How large were each of those groups? Were they roughly comparable, or were they?
No. So actually about half the sample was in the low steady group, and I would say, I think this early increasing group was pretty small. It was about 10%. And then I think the other ones were roughly around 20. Mm-hmm. Yes?
Did you know anything about their other drug use? Like maybe that early onset group switched to marijuana instead?
So I'm going to present the Arab cannabis results related to this. But yeah, I mean, their polysubstance use is really, really common in this sample. I mean, you see that kids who are using alcohol are also using marijuana. So we also saw an interaction between the internalizing polygenic risk score and disadvantage when comparing class membership in the early onset decreasing class and the low steady class. So specifically, individuals with a higher internalizing polygenic risk score, when exposed to higher levels of disadvantage, they're more likely to be in this early onset decreasing class. And so, again, sort of going back to what we were saying, we know in maybe disadvantaged communities it's easier to get alcohol. So if you're more likely, you want to use alcohol in early and middle adolescence for whatever reason, that might be the case especially if you're higher in internalizing genetic load and you have that predisposition to. And then, as I said, it's possible that in late adolescence, there's something going on. And there's some sort of repercussion or consequences that are, resulting in that decline in use that we see. So in sum, these results to me suggest that it's really important when we're looking at polygenic risk in relation to substance use that we really look across developmental periods, because we see that genetic risk at one developmental period could be associated with, risk for one outcome and not another. And they also highlight the importance of context as well. So now I'm going to sort of switch
Before
gears.
you go on, may I ask you a question?
Yes.
What that wasn't latent class analysis, was it?
It was a longitudinal latent class analysis.
And can you comment on the relative utility of that versus a trajectory under those things?
Yeah, so for this marijuana analysis that I'm going to discuss, we did do a growth spectrum, we did do trajectory analyses. When we were originally doing the alcohol analyses, we just had trouble getting some of the intercept and slope models to fit. So we just decided to do longitudinal class analysis just to make things a bit more streamlined. I don't think the results would really shift though, although it could give you
That's what I was wondering.
Yeah, I don't think the results would shift. It would give maybe a little bit more information in terms of intercept and slope, but I think we'd essentially be doing the same thing. So my current work is focused on looking at behavioral, environmental, and genetic correlates of opioid use. We got funding, it's a funding for a supplement to an existing R01. I'm really excited to present this work. So I don't think I need to tell this group that opioid use is a big problem. About 130 people each day die of an opioid overdose in the US. And in an effort to combat the opioid epidemic, a number of states have legalized marijuana. And the idea is that patients that are experiencing chronic pain, they would be able to substitute their prescription pain medication for marijuana, right? And there is some evidence that prescribing and mortality rates were actually lower in states that had these medical cannabis laws. Yet, about 32 states that have legalized medical cannabis have seen a significant surge in opioid overdose rates. So what's going on here? So it could be that cannabis is actually a risk factor for opioid use. However, there's a limited longitudinal community-based work in this area examining the relationship between cannabis and opioid. There's some evidence that cannabis before the age of 18 was associated with increased use of opioids between the ages of 18 and 25. There's also some evidence from animal and mice model studies that cannabis use enhances opioid intake and impacts the motivational and desirable properties of opioids. At the behavioral level, seeking out cannabis might result in affiliation with other drug-using peers, and it could just be that it's just easier to get opioids. We might see sort of this shared pathway. And so in the present study, we were interested in whether or not cannabis use across adolescence and young adulthood, whether that precipitated opioid misuse in young adulthood. So we used a subsample of the original sample that I've described, included about 600 individuals, majority African American, low SES sample. Marijuana use was assessed from ages 14 to 26. Opioid use was assessed starting at age 19 and continued to age 26. We defined opioid misuse as reporting using heroin or a non-prescription opioid anytime between the ages of 19 and 26. We were also interested in looking at whether or not cannabis was a risk factor for opioids, or whether or not that relationship was attenuated when alcohol and tobacco were also entered into the model, whether or not essentially those associations would go away. And so, very similar to what I found in the previous paper, we found 4 classes of marijuana use, a low non-users group, an adolescent onset limited group, again, same thing, to sort of increase in use up to age 17, followed by decline in use. We have a young adult onset group that sort of started using marijuana around age 19, 20, and then we have our adolescent onset chronic users. You can see that at age 14, they start using, and there's just sort of escalating. There's a bit of a dip in their use, I guess around after age 21, but you can see that they're still really using very much above and beyond everyone else in the sample. 15% of the sample, or about 90 people, reported misusing opioids. There were no significant differences in heroin or non-prescription narcotic use among the low/non-users and the young adult onset users. So the real differences occurred when we're talking about the adolescent groups. So relative to the low non-users group, the adolescent onset limited group was about 3 times as likely to use heroin, and the adolescent onset chronic users were about 5 times as likely to use heroin relative to the low non-users group. In addition, relative to the low non-users group, the adolescent onset limited group was about twice as likely to use non-prescription narcotics, and the adolescent onset chronic class was about 8 times as likely to use non-prescription narcotics. So, these results point, I think, pretty clearly to the fact that adolescence is a time of neurobiological plasticity, and it's a time when brain structures are developing in an enduring way, and the introduction of substances like cannabis, I think, could potentially really result in significant neurobiological changes that might make one more susceptible to using opioids in the future. There's also some evidence of shared morphology, right? So, for example, endocannabinoids and opioidergic receptors are found in shared neurobiological regions that are associated with reinforcement and reward. In addition, there's similar behavioral pathways and environmental exposures that might be associated with both cannabis and opioids. For example, being behavioral disinhibited, as well as affiliating with deviant and drug-using peers. So, sort of in sum, I think that it really points to adolescence as being a really important point of intervention when we're thinking about not just targeting cannabis use, but thinking about also use of other drugs in the future. So in the next study, we were really interested in, we saw this relationship between cannabis use and opioid use. Are we going to see that there's shared genetic risk as well? And so our first question was, is genetic risk related to cannabis positively associated with opioid misuse? And are the genetics underpinning other substance use outcomes in related phenotypes, such as risk for comorbid psychiatric outcomes, whether or not that's also associated with opioid misuse? So again, subsample original population I described, about 900 individuals, 80% African American. So I'm going to actually go through these sections really carefully, and I apologize if not the most interesting, but I want to hopefully make it clear why we saw the results that we did see. As you may recall, sample size and ancestry really matter. So we created 2 polygenic risk scores for cannabis. One focused on lifetime cannabis use, which included about 185,000 individuals, 72,000 cases, and all individuals of European ancestry. We also created a cannabis dependence polygenic risk score, included about 2,000 cases, and again, this was all individuals of European ancestry. We created 2 alcohol polygenic risk scores, one focused on alcohol use disorder and the other focused on alcohol consumption. You can see that it's a relatively large sample size and a mixed ancestry population. We created an opioid dependence polygenic risk score, which is a mixed ancestry sample as well, but you can see the discovery sample sizes are quite different. You have about 3,000 European ancestry individuals and about 1,000 African ancestry individuals. And last, we created a cross-disorder polygenic risk score. This was based on the PGC's cross-disorder working group. It's a meta-analysis of about 230,000 individuals of European ancestry. And essentially, we created a polygenic risk score. It was essentially a summed score of whether or not the participants had 23 loci that were associated with 4 or more psychiatric disorders, including schizophrenia, bipolar disorder, depression, etc. Okay, so just to kind of go through this table, we have heroin use here, non-prescription narcotics, and whether or not someone had reported heroin or non-prescription narcotics. And to the very left is, the polygenic risk scores. And in the yellow represents trend level significant associations, and the green represents significant associations p less than 0.05. And so what you can see here, the main finding is that the alcohol dependence polygenic risk score is predictive of non-prescription narcotic use, but not heroin use. And you can also see some other trend levels here with a cross-disorder PRS, which was also this trend level significant association for non-prescription narcotic use. What I think is interesting is, first off, you don't see a significant association with cannabis use, right? And it's, so you don't see a significant association with cannabis use. The results are very much driven by alcohol dependence, and they're really focused on, the results really hold just for non-prescription narcotic use. This is just a sample of some of the results that I've, some of the polygenic risk scores I've looked at. But across the board, we're seeing greater genetic prediction for non-prescription narcotic use than we are for heroin use. And so this suggests...
Including opioid dependence.
Yes.
That's really...
Yeah, could you go back to that for just one moment? Because I was trying to process that line of the slide.
Oh, sure.
That the opioid dependence PRS...
Yeah, so I just want to actually clarify this. So the way that I'll have to redo it, but essentially the reference group in what's reported is the opioid dependence group. So the odds ratio here, you can see it's below 1, and it looks like individuals with a higher opioid dependence score are actually at lower risk from heroin use. That's actually not the case. It's just the way that the discovery reports, the results were reported.
But it's not statistically
No,
reliable anyway.
not at all. Yeah.
That itself is very interesting.
Yes,
Yeah.
this was the African American. There was the African American GWAS, and then they did a trans ancestry GWAS as well. I ran that 2 days ago, and that I didn't include it in here, but that was also not
The
significant.
ancestry.
Mhm. Yeah.
And you used the African American summary statistics from Renato's Polomonti et al.
Yes,
And the target was African American cell.
a lot of times, that's, ideally you would have this same ancestry. I've sometimes used the trans ancestry findings because you can get more power when you, but that was not the case here. And so, to me, these findings suggest potentially different genetic etiological pathways between heroin and non-prescription narcotic use, particularly because we saw those sort of differences in findings. They also point to the possible importance of the genetics of alcohol dependence for predicting opioid use. I think that a very, very important reason why we're seeing differences in findings is because of differences in sample size and ancestral group of the discovery GWAS. I don't think that the genetics of cannabis and opioids are not associated with opioid use. That doesn't make sense to me.
I'm embarrassed to say I don't remember the sample size of Polomonti, but it's like about 40,000, wasn't it, the PGC?
Oh, the opioid dependence.
Yeah, right.
I mean,
No, it was a few thousand.
No.
It was like 1,000 or something, wasn't it?
yeah, about
was it?
1,200 African Americans.
Oh, okay.
Yeah.
Well,
Yeah.
that's fine.
It's,
Okay.
Yeah. So I think what these results, really suggest is it's very important to consider when you're evaluating findings, sample size, ancestry group. I do wonder if we had a larger sample size for some of these and more diverse ancestry population, if we'd see differences in the findings that we observed. So ideally, I would say, we could take all of these G/E findings and, actually put them in practice. And I do hope that one day they will be able to inform targeted screening and treatments that vary in terms of approach, timing, and dosage. But we are not yet at the point where we can do this for substance use behaviors and other mental health outcomes. Here are the roadblocks as far as I see it. I think many of the Genome-Wide Association studies, they focus on individuals of European ancestry. Another issue is that we're still determining what polygenic risk scores represent. I mean, what does it mean to have, we know that this allele is associated with an increased risk for disease, but what does that actually mean? What does that actually translate to besides this phenotype that we see? In addition, a lot of the G/E work has been conducted in small sample sizes, and I believe that in order to sort of make conclusions about the work, we really need large sample sizes, as well as replication in diverse samples. So from my view, how can we increase the diversity of genome-wide association efforts? I think one incredibly important thing to do is for academics and researchers and geneticists who are interested in this work, we really need to build relationships with diverse populations that may mistrust academic and medical institutions and have good reason to. We know that, I think everyone here knows about Henrietta Lacks and how her genetic information was exploited, stolen, exploited. And so I think that, it makes sense that certain populations have this distrust of, again, academic institutions and sort of genetic work, and I think it's our job to make sure that we develop relationships and develop trust among these groups. On a personal note, I'm a PI in a proposed partnership with a number of institutions in 23andMe. I am trying to get them to do a genome-wide association study on a number of substance use outcomes in their African and Latino ancestry sample. And the idea is that there's not many large-scale, diverse ancestry studies. So if we're thinking eventually down the line, developing these novel interventions and developing these novel treatments, we need them. And if we don't have them, it's going to contribute to the widening of already large health disparities. I am currently working on a KO1 And sort of my focus is on acquiring knowledge related to neuroimaging genetics, epigenetics, as well as data harmonization. Data harmonization, the goal of really pooling together as many samples as possible. And my goals are to really kind of parse out what the polygenic risk scores actually represent, to look at neurobiological and epigenetic mediators that influence polygenic risk and substance use relations, look at how environments influence those relationships as well. And I'm really interested in thinking about novel ways that we could actually predict substance use exposure, such as through teeth. Ultimate goal, I would like to move into translation and intervention work, although I think that there's so much work that needs to be done up front first before that can even happen. I think we can actually use a lot of this G/E work if we pull together individuals from multiple disciplines and backgrounds, develop relationships with all communities. And I think in doing this work, we have to be very vigilant in correcting misunderstandings and in our findings, and preventing our findings from being misappropriated, particularly in the current political climate. Acknowledge the grants that have supported this work. Hooray, NIDA. Thank you. Anyone have any questions?
So a lot of folks here are more knowledgeable than I am about PRS. On the table that you had, we were just talking about the opioid dependence, and I'm still chewing on. Those are correlations between opioid dependence and the likelihood of having increased non-heroin opioid use. Those are correlations.
They're associations. Mm-hmm.
Associations. So if you flip it the other way around, does it also say that these things are not it seems that there should be a stronger relationship between opioid dependence and opioid use or heroin use
Yeah, absolutely. So
Okay.
I think,
I'm just chewing on it, sorry.
Yeah, absolutely. I think I was surprised at this too, but as someone who's been doing this work for the last couple of years, sample size matters tremendously. And when you have a sample of 5,000, 10,000 people, it's just not enough. Hank was telling me that there was an opioid dependence GWAS that had about 80,000 individuals, 10,000 cases, and even then you only saw one hit. So when we're talking about a GWAS of opioid dependence that had 4,000 people, it's just not a lot. And then when you take into account the ancestry piece,
Okay.
I'm heavily involved in creating polygenic risk scores, and when you try to apply predominantly European ancestry GWAS to another population, you lose a ton of SNPs. You lose a ton of SNPs that might be relevant.
Right.
Mm-hmm.
Got it.
Yes.
So, beautiful work, and it makes me stimulated, to think about other studies. And it would be really great if we had biomarkers in the mix. You reviewed a lot of different studies using the genetic risk factors, but there's a big literature on inherited tolerance, essentially, reactivity. Has anyone combined the biomarker approach with the genetic approach in the longitudinal way that you described?
So can you speak to, like you said, inherited
Tolerance, like Mark Shuckett is one of the ones who's done the most in this area, but others have too. And essentially, there's a simplified description of it is that people who have high tolerance, in other words, low reactivity to alcohol, is easy to test. That they have a increased risk for developing alcohol use in fairly young age too. Under 30, I think.
Okay.
For the most part.
I don't know if anyone's looked at that specifically, but that would be really interesting and I think informative to think about and potentially pursue.
think you could probably, since COGA, Collaborative Study on the Genetics of Alcoholism, has phenotyped that Mark is part of that consortium.
Okay.
And they've GWAS'd it. You probably can get the summary statistics for that familial risk, the tolerance-related risk. It would be, so it's an empirical question that could be answered.
Absolutely.
I think another thing is there are really large samples of smoking from GWAS.
Yeah.
Really large samples.
Yeah, there was a paper that came out, the last name Liu, L-I-U.
Yeah, in December, yeah.
Yeah, we just got the
1.2
23andMe
million, yeah.
summary stats, so we were waiting on those.
And alcohol use, those are
Yep.
the two phenotypes, yeah.
So where you were presenting the cannabis PRS and the opioid PRS and so on in that last big table,
Yes.
do you have measures of those like primary phenotypes? So can you test to see, does the cannabis PRS actually associate with cannabis use prior
Yes.
to whether or not it associates with narcotics?
Yeah,
use?
trying to think if we've done that. I know we were doing that, we did that in a different sample, on a different set of results. But that is something that we do typically do. And that's something that I've struggled with as well, because if you create a polygenic risk score we're supposed to reflect risk for cannabis, but then it's not associated with cannabis use in your sample, then what is it
Right,
actually
well, yeah,
represent?
then
Those
it might
underlying-
mean that it's just not a good PRS, right? If it's either because of the ancestry or because of the sample size and so on.
Yeah, so with our internalizing polygenic risk score, we actually did not see very strong relationships with anxiety or self-reported anxiety and depression. But what's interesting is that we believe that people underreported their level. I mean, the anxiety and depression was almost zero in the sample. And I don't think that we're talking about a really low SES population that's exposed to a lot of significant stressors. I can't imagine everyone basically saying that they're not anxious or depressed. So then it's like, well, maybe in the internalizing score isn't terrible, but maybe people are underreporting, or maybe the measurement of of internalizing isn't really good. So it's tricky, but I think that that's a great idea. Yeah.
There are a number of people here from the PET Center.
Oh.
And so I just want to clarify that what we're doing is based on that GWAS of 80,000 people. And we've generated PRS from those summary statistics in the sample that's undergone carfentanil scans that young Car Jubieda has. And so we have a nice large derivation sample and a comparatively large target sample of people who had PET. And so we're hoping to refine that.
Mm-hmm. Yes.
So you mentioned partnering with 23andMe and for an African and Latino heritage study and everything like that. I was wondering, I mean, you were talking about partnering with communities that are reluctant to start and everything like that. So what is that looking like as you plan for that particular GWAS or?
Yeah, so I guess there's two pieces. Like the first is so 23andMe has a really large sample size, and the idea would be there's not a lot of, so there's a GWAS that Hank had mentioned that's really focused on alcohol and tobacco use. Everyone in the sample is of European ancestry. So the idea would be to ask 23andMe / beg 23andMe to please do these genome-wide association studies on alcohol and tobacco use in their samples in these populations.
So already collected.
Correct, yeah. I think, just to be frank, at this stage of my career, I mean, I don't see myself going out in the community. I mean, I think, and trying to get people to donate DNA, I think from more of a broader perspective, that when, for individuals that are collecting data on participants, that it is really important that we make sure that we're not just targeting samples that are easy and accessible, and we're making sure that we're targeting populations that it may not be that way.
Stella, you want to comment on that?
No comment.
I have a question for you. How often does letting them know, obviously your whole studies are genetic, how often does that deter them from wanting to actually be in the study? Like, are they saying, oh, I don't want to give my genes up at all, or like, I
I'm
don't
not
want to
sure
be
what
in
you're
the study?
asking.
Like, knowing that they have to give you their genetic info and they're getting a blood draw, does that deter them from wanting to do the study
It
ever?
happens.
A lot, or how much? 'Cause
I'm not particularly often, usually it's.
But we advertise, I mean, it's clear up front,
okay.
so there may be upfront self-selection, there is upfront self-selection.
Right, many times they think that they are getting like a 23andMe readout from the study, and when we tell them, no, you can't see your individual results, then they might say, no.
Oh, interesting.
Yeah.
Tell them they can help
And-
capture a Golden State Killer or something.
Right, exactly,
Do you
yeah.
have any idea how large the African-American sample is, or the Latino sample is in 23andMe?
So they have advertised online that their I don't know if I believe it personally. They said that their African American sample is like 800,000. Their Latino sample is about 200,000 or 300,000. The question is whether or not they also have phenotype data. That's the thing,
Yeah.
because
That's marketing, I mean, this is the realm of marketing.
yeah. I would-
The other question is, how valid is the self-report on 23andMe?
Well, so that's a really great question. So I don't, well, there's two pieces. The first is, so I actually signed up for a 23andMe kit, mainly because I was trying to figure out what the heck, the questions at what were the actual questions that 23andMe had asked participants. So I signed up for a thing. I didn't even send it in. I just wanted to know the questions. But, you know, it really takes a type of person. I mean, I was sitting on my computer. I spent 3 hours filling out these questionnaires.
Oh my gosh.
Oh,
I
it'll
know.
keep going, and it'll follow up.
Yes,
with
yes,
additional-
yes.
As after you first submit materials, they'll send you, if you endorse a willingness,
Yes.
continue
Oh,
Yes.
wow.
I still
They'll
get
keep
emails. Yeah.
pumping you for information.
Wow.
Yeah.
Yeah.
Of course,
I didn't know that.
people with substance use issues might be less likely to
Yeah, exactly. Or
answer
Yeah.
those.
even if you have other things to do. I mean, I was motivated to do this.
Yeah.
But so if you think about like, who are the people who are actually taking the time to do, and people like me, oh God, you know, but you know, it's people who are potentially, you know, sort of hiring conscientious who really want, you know, those data. So I think that's one piece of it. But then the second piece is, you know, 23andMe, I mean, a lot of times it's completed by higher SES individuals. I mean,
Yeah.
you know, so, you know,
And they're not doing it for the public good. They're selling this to pharmaceutical companies and, I mean, they're monetizing people's data. I did 23andMe because I was curious too.
And you have to release it to all those people?
You sign, yeah, it's
You can choose.
anonymized.
Yeah, but you
Yeah.
can opt
Choose
out.
not to, yeah, you can choose not to do a search.
That's kind of why I was asking, like, if you've used the example of the HeLa cells and everything like that, and then you go with like with 23andMe, a place that is monetizing the data, like the data that they're mining and everything like that, it seems there's a lot of space for some uncomfortable overlap with that.
yeah, I mean, they tell you they're gonna do it though, when you sign.
Yeah, they
Yeah.
have to be upfront.
Yeah.
But also low SES people aren't gonna prioritize buying 23andMe, so they can't afford, you know, it's
Yeah,
expensive
but
even to
even-
just do it if maybe
It
if
is.
they.
But
Internet access.
yeah, but SES isn't the same as Ancestry, right? Like even if
Yeah, but if they can't afford to buy the kit, I think that if you can't afford to like buy food, you're not gonna buy a 23andMe
right,
kit.
certainly,
Yeah.
yeah, certainly.
We should probably stop,
Oh yes.
continue informing.
Thank you.